So in today's lecture we'll discuss pluripotent stem cell models to study neurodegenerative diseases. And briefly, the outline of what I'm going to say today, I'm going to give a brief introduction about embryonic stem cells and pluripotent cells, to talk about disease models of embryonic stem cells and why we need disease models of embryonic stem cells, especially for neurodegenerative diseases and neurological disorders. I'm going to discuss other means of making pluripotent cells and maybe talk a few minutes about the ethical issues that underlie stem cell research, embryonic stem cells in particular. And the two means which we're going to focus on are cloning, somatic cell nuclear transfer, and reprogramming, which is gaining more and more momentum on a daily basis. Okay, so let's start by introducing the concept of embryonic stem cells, what these are, and what we can do with those. So here you can see the early stages of development. This is a fertilized egg, and after fertilization, the egg starts dividing. It divides into 2 cells and then into 4 cells, as you can see here, 8 cells. So this takes a few hours. Okay, so in a matter of a few hours, the cell divides into 4 cells, 8, 16, 32, 64. No, so about 64 cell stage we stop counting. Not because we can't count more than 64, it's because that the divisions are no longer synchronous, okay? So, and you don't really get a 128 or so stage. What you do get is a pack of cells, a ball of cells, which we call morula. And this morula increases in size and cell number, and after about 5 days, 4.5 days, in humans, it turns into what we call a blastocyst. So in mice, we're talking about day 3.5, and in humans, day 4.5, okay? Now, the major difference between the morula and the blastocyst is that the blastocyst includes a cavity, okay? So this is hollow, and you have 2 cell types in the blastocyst. Unlike the morula, which we can't really differentiate the cells, they're all sort of the same cell type. Okay, so these are just dividing cells from the original fertilized egg. Here we see 2 kinds of cells. The outer cells, which are the trophoectoderm, and these are the cells that will become the placenta. Okay, so these are non-embryonic cells. And these guys here are called the inner cell mass. Okay, so there's an outer trophoectoderm. This is hollow, as you remember, and these guys are the inner cell mass. These cells will become our bodies, okay, will become the embryo. So these cells have the potential to differentiate and become every single cell of the human embryo, or the animal embryo, depends on which species we're working on. And if we can culture these cells, if we can take these inner cell mass, so here's is the same 3D image of the embryo. This is, you can see the trophoectoderm in blue, the inner cell mass in orange. And if we take these inner cell mass and we culture them in a dish, we have generated what we call embryonic stem cells, okay? So embryonic stem cells are an in vitro manifestation or in vitro artifact, if you want, of these inner cell mass. And I just have to spend 1 minute on the difference between embryonic stem cells and these real embryonic cells, because they're very big differences. These cells here are very transient during development, okay? So the embryo keeps on developing. Immediately after this stage, the embryo transplants in the uterus, and then gastrulation begins. So all the formation of all the different organs with firstly the formation of the central nervous system, and then the formation of the organs. So they only have a few hours that they are these inner cell mass or these pluripotent cells. But if we successfully culture them, we basically have this immortalization stage where these cells self-propagate indefinitely. So if we culture them successfully and we derive an embryonic stem cell line, these cells will now propagate as many times as we want them to. Okay, so they can divide and divide and divide, and if we let them, they can fill the planet. We usually grow embryonic stem cells on top of feeder cells. You can actually see them here in blue. We usually use mouse embryonic fibroblasts as a mat or a scaffold just to grow the cells on. So these fibroblasts secrete factors that help embryonic stem cells grow and derive. And again, when they grow successfully, we can say that we generated an embryonic stem cell line. Now, importantly, we can mimic the early stages of development in the dish. Now, if we give them the right conditions, we can now differentiate these cells into early embryoid bodies, so-called embryoid bodies, which mimic quite nicely the early stages of development. So it gives us a glimpse into early human development, which we cannot do in real humans. So the two main characteristics of embryonic stem cells are, firstly, that they can self-renew indefinitely. It means that one embryonic stem cell becomes 2, 2 become 4, 4 become 8, and so on. And they are identical to each other, okay? So we can, as I said, we can let them self-renew indefinitely or almost indefinitely. There's an issue about how many times we can let them divide, but this is not the point. The point is that we can generate many, many identical cells, and we're talking about billions and trillions of identical cells, which are undifferentiated. However, the other very important characteristic of embryonic stem cells is that when we give them the right conditions, the right cues, they can now differentiate. So at some point, I can take some cells from a dish, put them in a new dish that is coated with some magic substance, and I add a little bit of salt and pepper and paprika and whatever, and lo and behold, they can turn into a neuron or an astrocyte or a liver cell or something else. Now, how to differentiate these cells into the different cell types is one of the major, I would say, challenges in embryonic stem cell research and in the future for the clinic, because we really don't know. I mean, it's a lot of trial and error. People, many labs are trying to derive specific cell types. And there is a growing list of, protocols, cookbooks, how to derive a dopaminergic neuron, how to derive a blood cell or a B cell or a T cell, and so on and so forth. So far, we know about maybe somewhere between 50 and 20 cell types. There we have another 220 to go. Okay, so there is still a lot to be learned in that particular field. But still, there's a lot of excitement because we can do it. So in essence, these cells are very beneficial for therapy, especially for diseases in which cells die, right? So the classic example are neurodegenerative diseases such as Alzheimer's disease, Parkinson's disease. Cells die. Cells die in the brain, in the central nervous system, and to replace those cells, to replenish the affected tissue, we can use embryonic stem cell derived neurons, for example. Okay, so this is the major excitement that we have about replenishing organs using embryonic stem cells, because we can generate as many cells as we want. Okay, but in addition to embryonic stem cells, which, as I say, are pluripotent, meaning they can generate every cell type in the body. There are other ways of making pluripotent cells, and this is by reprogramming differentiated cells or somatic cells back to pluripotency. And I'm going to spend a few minutes discussing the different modes of reprogramming. How can we do it? How can we take a differentiated cell, a somatic cell, and turn it back into an embryonic stem cell? But before I do, I want to touch on some ethical issues and experimental paradigms that have been developed in the past years, and ask, can we actually generate human embryonic stem cells without the destruction of embryos, and can we generate disease models? So for that, do we have, no? Is it بلتي مخيك? تمام. Thank you. Okay, so this is the human embryo. Here is the ICM. When we derive embryonic stem cells, we are actually destroying this embryo. Okay? And of course, in humans, when we derive embryonic stem cell lines, we are not taking embryos from women, from actual humans. We are only using embryos derived by in vitro fertilization, IVF. Okay? So a couple goes to the clinic, they can't get pregnant for any reason, and they go to an IVF clinic. The woman gets a series of hormonal injections so that she will produce many eggs, as many as possible. And then in the clinic, they fertilize the eggs. Okay? So this is in vitro fertilization. So now we have a tube with several fertilized eggs, early embryos. So what they do in the clinic is they take these embryos and they now differentiate them into during the first stages of development. Okay? And then once we have these embryos, we can transplant them back in the womb, in the mother, and hope that she will get pregnant. Okay? This is the procedure of IVF. Now, let's say I started with 12 embryos. I put 3 back in the woman, and the rest are frozen. They are frozen usually for 10, 20, 30 years, but then discarded. Right? The woman is now 50, 60 years old. There's nothing to do with these embryos, and they're normally discarded. Instead of discarding these embryos, they can give them to research, which means that now we can take these embryos and try and derive embryonic stem cells from these embryos. Okay? Of course, we are destroying the embryo, but this is an embryo that would have been destroyed anyway. I mean, it's an embryo that was kept frozen, and the couple doesn't need it anymore, either because they already have the number of children that they want, or that they're now at an age that is not relevant for this. So these are embryos that would have been discarded, and they can be used for research in most places. In most places in the world, they can be used for research. There's a whole debate about this in the US, and for many years, the taxpayers' money was not used to derive human embryonic stem cell lines from discarded embryos. The belief of I guess most of the population in America, or maybe not most, I didn't really conduct the poll, but there is a sentiment in the US that it's better to discard the embryos rather than derive embryonic stem cell line, because then you destroy them for research. Okay, so we can use these lines, but we as I said, by doing so, we are destroying the embryo. Okay, this is a given. So the question is, can we generate embryonic stem cells without the destruction of the embryo? And the answer is yes. And the procedure is called pre-implantation genetic diagnosis, or PGD. Okay, so now let's go back to the same couple that is undergoing IVF, but they go to the IVF clinic not because they cannot get pregnant, but because one of them is carrying a mutation. Okay, so let's say I'm a carrier of the Huntington disease mutation. This is a mutation. Well, I'll go back to it in a while, but just as a teaser, this is a mutation in a gene called Huntingtin. It's a polyglutamine mutation, which means that instead of about 15 glutamines, glutamine is designated as Q. So it's about 15 in healthy individuals, and in the mutated genes you get about between 50 and 150. Okay, so it's called polyQ, polyglutamine expansion. Now, if I carry this mutation, I am going to develop the disease, 100% sure, unless I die earlier. So this is a late onset neurodegenerative disease. It appears around the age of 40, 50, 60, depending on the length of the mutation. Now, if I do carry such a mutation, I know that I'm going to develop a disease, but I don't want my child to suffer the same, the same result. So what I can do is go to the IVF clinic with my wife, undergo the IVF procedure, but now we're going to take a single cell from each of the embryos before we transplant them back. Okay, so let's go back here. These are the small embryos. Now we are going to differentiate or allow the embryos to develop only to an 8-cell stage. Okay, so we have the fertilized, this is the fertilized egg, oocyte, and then we have an 8-cell stage embryo. So we do it to all the embryos that we have. Okay, so these are the embryos that we had. We are taking a single embryo, a single cell from each embryo. And now we can screen for the mutation. We can do a PCR test. There are other tests that one can do. We can screen for the mutation. And then find out that we have, let's say, 2 mutated embryos and 3 healthy embryos. Okay? And now we can put the, it's fine, I'm not going to use it too much. It's okay. So now we can put the embryos, the healthy embryos, back in the mother. And at the same time, we now have embryos carrying a mutation that we can derive embryonic stem cells from. And this is a very, very powerful tool, because we have cells, human embryonic stem cells, which carry the polyglutamine mutation or any other mutation that is available. And we can actually use them to study the disease. We can now differentiate the cells into neurons, study what happens to the neurons, because neurons are essentially impossible to get. So essentially what happened here is that we generated embryonic stem cell lines without the destruction of the embryos, because we were able to take a single cell and the 7-cell embryo transplanted back, or actually let it develop first into a blastocyst and then transplanted back. So apparently this procedure does not affect the embryo, luckily, and I think the kids that were born, firstly born by the PGD procedure, are now around 7 years old, and they're all healthy. So it seems to be working properly. Okay, so you can take a single cell from an 8-cell stage embryo without harming the embryo. This is important. So this cell that we took from the mutated embryos, we can, so we did this mutation, but these embryos that carry the mutation, so we have one cell less here. Now what we do is we allow it to differentiate into a blastocyst. Okay, this is the inner cell mass, and we take the inner cell mass and we derive human embryonic stem cells. Okay, so this way we can generate embryonic stem cells which carry a genetic mutation. Okay, so as I said, we're going to discuss means by which we can reprogram cells back to pluripotency. And historically, the first way that people showed that this is actually possible is using somatic cell nuclear transfer, SCNT, or as most of you know this procedure as, cloning. Okay, and I'm going to go back to it. I'm just going to present the methods first. The other method that was developed is through cell fusion. And the idea is if you take an embryonic stem cell and you fuse it to a somatic cell, the somatic cell turns pluripotent. So the fusion renders the somatic cell pluripotent, and we'll discuss it a little bit. It's not very helpful. I mean, it's not helpful at all for therapeutic purposes, but for basic research, it's a very, I think, important paradigm. So the other means, which is basically not working properly, but working to some extent, is using extract. So here, for example, in somatic cell nuclear transfer, we are transferring a somatic cell into an egg. And the egg itself, the oocyte, has the power to reprogram, has all the factors required to reprogram this cell into a pluripotent cell. It erases erases the memory, erases all its identity, and turns it back. So it means that we have factors inside the egg. So what if we take now 1 million eggs, we put them in a blender, and we just add them to the somatic cells, right? This is technically possible, and it does help. So if you do this experiment, you can see that with extract you can differentiate, but you can reprogram cells, but not all the way. So you need something else, but you can, this is just, we're not going to discuss it, but it's maybe important to remember that maybe with extract you can help reprogramming or at least get partial reprogramming. And finally, the most exciting way that has been recently developed, the Yamanaka way. So Shinya Yamanaka, a Japanese researcher, published about 5 years ago a seminal paper showing that by introducing 4 transcription factors, and later 3, OCT4, SOX2, KLF4, and c-MYC, the names don't matter, into somatic cells, fibroblasts, the cells reprogram into cells that look just like embryonic stem cells. They are called iPS cells, induced pluripotent stem cells. In fact, this Sunday, just 4 days ago, Yamanaka, together with Yenish, received the Wolf Prize from the Israel president and prime minister at the Knesset. This is the most prestigious scientific prize given in Israel. And if I read the map correctly, in the next three years or so, he's going to get the Nobel Prize. So this is really a major breakthrough. Why? Because now I can take a skin cell from each and every one of you and reprogram it to become an embryonic stem cell-like cell, and then differentiate the cell into a neuron or a liver cell or whatever. And if you're, God forbid, sick or have some kind of degenerative disease, I can transplant it back in your body and recreate your missing organ, right? Without any problems of rejection, because the original cell was taken from your own body. So the implications of this reprogramming procedure is immense. Okay, so let's go back now and talk a little bit about somatic cell nuclear transfer or cloning. So the idea behind the cloning procedure was developed by this guy, John Gurdon. And again, if I read the map correctly, if Yamanaga gets the Nobel Prize, he's going to share it with John Gurdon, who was actually the first one to show that reprogramming is possible, that erasure of identity, of cellular identity is possible. So John Gurdon used this African frog called Xenopus levis. And the reason he used this model organism is because these frogs have very, very large oocytes. You can see this is 1 millimeter. So the oocyte is bigger than 1 millimeter. You can see it with your own eyes. You don't even need a microscope to manipulate these oocytes. So for experimental purposes, it's a gold mine. So he mated these frogs, as you can see here, their favorite position. And then what he did is after he fertilized the oocytes, he removed the nuclei. Okay, so now he has an empty oocyte with no genetic material, no DNA, no nuclei. He then went back to the frog. This is an enlargement of the gut. And he took one single cell from the intestine, from the gut. And he injected it back into the oocyte. Okay? So the oocyte has to contain 4N genetic material. 4N is every cell in our body contains 4N because we have 2N from our mother, 2N from our father. And this is exactly what this oocyte contains. Okay? He removed the 4N, all the genetic material, and he put in the equivalent in terms of genetic material. Okay? So in terms of the genetic material, the oocyte has exactly what it needs. Only, instead of having this 2N from the father, 2N from the mother, it now has 4N from the frog. Okay? Father or mother, it doesn't matter, or a different frog altogether. And the question is, what happens if you now will take this oocyte and put it back in the frog? Will it develop properly? And in again, another seminal paper which was published 1962, John Gurdon was able to show that reprogramming is possible, and he got these tadpoles. These tadpoles never made it into frogs, okay? But nonetheless, it was the first demonstration that you can get a live offspring by cloning, by somatic cell nuclear transfer. The first one who actually achieved a whole animal was a Chinese researcher, the name of Tong Dizhu, and in the mid-60s, he generated a carp, carpion, by somatic cell nuclear transfer. But when people tried to repeat these experiments in mammals, it didn't work. It didn't work. We're talking about 1960s, and there were attempts in the 60s and 70s and 80s, 30, 35 years until Dolly. Dolly was cloned in 1996. So it took another 30 years or so to clone a mammal. It was successful. There was a whole debate about Dolly, whether it's really a somatic cell or not. We'll touch upon it a little bit. But the idea was similar. They took an oocyte from a sheep, enucleated it. Enucleated means they removed the nucleus, and inserted the nucleus of a somatic cell. Again, in terms of genetic material, we have exactly what it takes to make a whole animal. Okay, so you put it back, you put the fertilized egg with the introduced nucleus back into the foster mother, and you get an identical clone named Dolly. This cell, this somatic cell, was from the mammary gland of the sheep. And this is why the Scots named it Dolly. So it's some kind of Scottish humor, right? Because Dolly Parton was known for her big mammary glands. Right, so, and then the race began, right? We know that it's possible to clone a mammal. By the way, it took them hundreds and hundreds of attempts. And there were a few tricks that they did with electricity. I mean, it wasn't that easy, but it worked. It showed the world that this actually works. You can clone a mammal, and indeed, it started a race to clone other mammals. Here you can see a cloned cow in 1999, and the importance of this cow is that it was the first mammal clone to produce a viable offspring. Okay, so the clone produced a viable offspring. By and large, all the cloned animals are not very healthy. They usually die prematurely. Nobody really knows exactly why, but still there is there are attempts to clone animals, and some of them are endangered animals. So I think that the cloning procedure will enable to keep some of these endangered animals alive. So here's an example. This is Dewey, the deer. This is Snoopy, the dog. This is not a spelling mistake. This is the way they called this dog Snoopy, which is not the spelling of the real Snoopy. And I'm going to use Snoopy to give you an estimate on how difficult the cloning procedure is. So here's what they did. They collected 2,000 eggs, and they had 1,095 successful fertilized eggs, okay? So you take 2,000 eggs, you try to fertilize them, and you generate early embryos to put back, okay? So already here in this procedure, they lost half of the embryos. But they did generate 1,095 embryos. They transplanted these embryos into 123 dogs. Why? Because these dogs, they have 8 pups or so. So the litter size is around 7, 8 to 9, and therefore they took the same ratio, the same litter size, so it'll be as accurate as possible. So they used 123 dogs for this experiment, and they only got 3 pregnancies. Only 3 pregnancies, out of which only 2 live births were recorded, and only 1 surviving clone. So you can see the ratio here is quite disappointing, and this is the reason that cloning is still very difficult and cumbersome, and we don't really know why it doesn't work. But the efficiency is still very, very low for most animals. So here is a Snoopy, by the way. This is the clone. This is the father, or the dog that gave its single cell to generate Snoopy. Yeah.
So the DNA will be exactly the same.
Yeah.
It will be
Yeah. So they are identical. There are epigenetic changes, which maybe I'll have time to discuss later, but genetically, they are identical. Here's another example of wolves that were cloned in Korea in 2005. These are another example of endangered animals. And here's a variety of different animals that were cloned. You can see pigs, rats, mice, donkeys, sheep. Dolly died in 2003, quite young for a sheep, actually. This cat is called Copycat. And about two years ago, a little less than 2 years ago, a camel was cloned in Dubai. Now, why? Because apparently there are camel races in Dubai, and a fast-running camel is a big money-making machine. So they actually took one of the fastest camels in Dubai, and they cloned it, and this is the clone. So we're now waiting to see if this clone will run as fast as its father. So if so, you know, we'll be able to, in the future, make money from cloning animals. Okay, so well, yeah, there are some. There were some attempts, none that worked. No, not in mammals anyway. So in humans, cloning is forbidden in all countries in the world. However, what you can do and what is allowed, again in most places, not in all places, is what called therapeutic cloning. And therapeutic cloning is cloning for the purpose of therapy, which means that for the purpose of generating embryonic stem cells. How do we do that? So the idea is the same. You do exactly the same procedure, but now you start with a human egg. Okay? So you start with a human egg, you fertilize it in vitro. You take a human cell, somatic cell, from you or you or you, and you put its nucleus inside this fertilized egg. You, again, in vitro. Everything is in vitro in humans. You allow it to divide, and you generate the blastocyst, the embryonic stage from which we derive embryonic stem cells. And then we can do this. We can take this human embryo, take these cells, and generate human embryonic stem cells. Now, again, this is very powerful because I can generate, if this is working, in theory, we can generate human embryonic stem cells that are identical to you and you and you and you, okay? Just like the Yamanaka way. And indeed, so what's with human cloning? In March 2004, a Korean group published this paper which appeared on the cover of Science, evidence of a pluripotent human embryonic stem cell line derived from a cloned blastocyst. This was the first successful attempt of human cloning. Okay, so basically, of course, you cannot proceed after the blastocyst stage, but they reported that the entire procedure in humans worked. They took they took an egg, fertilized it in vitro, took a fibroblast, put it inside the egg, let it develop into a blastocyst. A year later, the same group, not only did they manage to do the initial procedure, but here they basically report that they produced patient-specific embryonic stem cells from human somatic cell nuclear transfer for human cloned blastocysts. So here what they've done, they took 11 lines. They took cells from 11 or 10 individuals. You can count it, I can't remember. And again, the cover of Science reported these findings. And they claim that they now have, sorry, just my only watch, so I have to keep it in. So they took cells from 11 patients or 10 patients, and okay, I have to shut it off. Sorry. They took cells from 10 patients, and they derived embryonic stem cells through in vitro fertilization, generation of blastocysts, and generation of human embryonic stem cells. So now they have patient-specific embryonic stem cells. They can study the disease or whatever, generate liver in vitro and so on and so forth. Unfortunately, this was all fraud, and in December 2005, a few months after the second paper was published, this broke out, the whole story broke out that basically they just lied. And it's quite silly, really, because it's the easiest thing to do, to check the validity of these cells. And of course, people have done it, and this guy, from being one of the most famous scientists in the world, became a big loser. Actually, between June 2005 and December 2005, I had the pleasure or the non-pleasure of hearing him in a small conference. He was really accepted like a half god, but that's the way it is. Okay, so we talked about somatic cell nuclear transfer and the way that, so just as of now, this has not been achieved in humans. Okay, so we're back to day one. So we talked about somatic cell nuclear transfer, and I'll spend a few minutes talking about fusion. So the idea behind fusion came from experiments by Helen Blau and other researchers, which basically show that when you fuse two different cells from two different developmental stages, the early developmental stage takes over. It means that if you fuse embryonic stem cells with somatic cells, the earlier, the ES cells, will take over. And you can actually screen for pluripotent colonies, and this is how the fusion looks like. So you basically, here's the fibroblast, here's an embryonic stem cells. This is the first stage, they first fuse, and then you can wait a few days and look at colonies that appear morphologically just like embryonic stem cells. Now these colonies, for example, they express a green fluorescent protein under the regulation of a gene that is only expressed in embryonic stem cells. Okay, but this gene, this green fluorescent protein, is in the genome of the fibroblast. Okay, so you have a fibroblast, these fibroblasts right here, which you can't see because they're not green, they all have this green fluorescent protein, only it's not expressed. Okay, but when these fibroblasts turn into embryonic stem cell-like cells, or when the fusion is successful, they suddenly start expressing this green fluorescent protein, become green, and then you can see these green colonies. So this is just a nice readout to see the reprogramming process. So these are actually fibroblasts that have become pluripotent. Now there's a problem with this procedure, because when we fuse two nuclei or two cells together, we get double the amount of genetic material. So this is 4N. Here, if you count it, you will see that there are 40 chromosomes. This is in mouse cells. So mice have 20 chromosomes. 1N is 20, 2N is 40. So here there are 40 chromosomes, but in the fusion there are 80 chromosomes. So of course, we won't be able ever to use fusion cells for therapeutic purposes. We cannot use tetraploid cells. Tetra means that we have double the amount of genetic material. But again, as I mentioned earlier, it's used for basic research too. So that's just what I wanted to say about fusion, and I'm not going to say anything more about extract more than what I said earlier. So now we're going to go back, and I'll show you the Yamanaka experiment. Okay, so this is an experiment that was published only 5 years ago in mouse, 4 years ago in humans. It was published in Cell by Takahashi and Yamanaka. And here's what they've done. It's a very naive experiment, and this is why it took time for people to believe them, especially what happened after the Korean scandal. And in fact, these were terrible years for stem cell researchers after Wang Suk faked the results, because every paper that was sent for scientific journals from stem cell researchers was just looked at with 17 different reviewers, and every, they were very, very afraid that another thing like that would happen. So what they've done is they looked for proteins, transcription factors, that are highly expressed in the S cells. And their naive idea was, if we take these proteins that are highly expressed in embryonic stem cells and we express them, we force their expression in somatic cells, maybe these somatic cells will somehow miraculously turn into embryonic stem cells. So they ectopically express them in somatic cells, fibroblasts. And then, and this is the tricky part, because people have tried this before, they actually waited several weeks. Okay, so from somatic cell nuclear transfer experiments, cloning and fusion, we know that reprogramming is not immediate, but it doesn't take weeks. It usually takes days, maybe 1 week. But they actually had they actually waited for several weeks. It took them 3 to 4 weeks, and they managed to get embryonic stem cell-like colonies. So they started with 26 factors, and after they saw that this procedure is working, they narrowed down the list to 4 factors, as I mentioned earlier: OCT4, SOX2, KLF4, and CMYC. So this was really amazing. You can take these 4 proteins, put them into any cell type that you want, and lo and behold, it turns pluripotent. And in fact, these things were called iPS cells, induced pluripotent stem cells, and they look exactly like embryonic stem cells. They behave like embryonic stem cells. They differentiate like they do exactly the same. There were some problems with these, and I will talk about them, but the early days were quite nice. So this is just a figurative image of this experiment. They took these 4 proteins, or genes or proteins, they inserted them into a fibroblast, and these fibroblasts turned into these so-called iPS, induced pluripotent stem cells. You can see the colonies right here, which are essentially similar to embryonic stem cell colonies. Okay, but there are major problems with this technique. First of all, one of the 4 factors, CMYC, is actually an oncogene. It's a gene that causes cancer. That's a big problem. Second, they actually used viruses in order to express these proteins, to express the genes inside the fibroblast. They used viruses because viruses are very efficient in expressing ectopic proteins. So of course, there's no FDA that will allow to use cells that they have viruses. Thirdly, these viruses integrated into the genome of the fibroblast. So again, we don't want any genome manipulation if we are to use these cells in therapy. Also, the efficiency was very, very low. About 1 in 10,000 cells was successfully reprogrammed, and it took time. And also, there was a problem to make whole animals. So with embryonic stem cells, with mice embryonic stem cells, for example, you can put them back into a pregnant female, pseudo-pregnant female, and she will, the embryo will develop properly, and you can get pups. You can get animals from embryonic stem cells. With iPS cells, in those early days, 2006, 2007, it wasn't possible. You could get chimeras. I'm not going to explain exactly what this is, but you couldn't get whole animals, animals that have their entire body made from iPS cells, only some parts of their body, okay? So, in fact, today, 5 years later, there's solutions almost to every problem. So the first problem was that CMYC is an oncogene. It causes cancer. And indeed, all the mice, the chimeric mice, which were derived from iPS cells, or some of their cells are iPS cells, they all developed tumors. So this was, of course, a major problem. So the solution was to try and do the reprogramming without this factor, and it worked. So it took longer, about 1 week longer, and this is why they never came out with this in the first place. But when you now know that this works and you can try it without CMYC, it works. The problem now is that the efficiency is even lower. So if it was 1 in 10,000, now it's 1 in 100,000. It works with lower efficiency, but you know, the cancer is a much more problematic than low efficiency. Okay, so integration into the genome. The solution is actually you can use non-integrating viruses. Okay, for example, you can also use plasmids, proteins, RNA, things that will not integrate into the host genome. I also said that use of viruses is a problem to begin with, so these are more or less the same solutions. Again, you can use DNA that is non-viral DNA just to express the genes without the virus. You can use genomic elements that are non-viral. You can use the protein itself and not the DNA, and you can actually use RNA as well. So again, all these procedures have been shown to work. The efficiency varies. Usually it's very, very low efficiency. Viruses have the highest efficiency, but again, if we are to go to therapy with these, we have to take measures of security. So the low efficiency has not been really solved yet, but some groups have shown that you can improve the cocktail. You can add genes such as this nuclear factor or this gene, and you can improve it. The improvement has not been dramatic so far, but there are some drug companies, of course, that are screening for compounds to try and improve the reprogramming efficiency. In fact, there is a company at Harvard that showed that they can replace each of the 4 factors with a drug, with a small molecule, which is, you know, it's very nice. You can use, you know, drug 1754 instead of OCT4. You can do this, but when you combine the 4 drugs together, nothing happens. So they're still trying to get there. But if reprogramming will be possible only with drugs, this will be a major breakthrough. Also, there are some chemicals and drugs that were shown to improve reprogramming, such as histone deacetylase inhibitors, and I'm going to go back to this family of drugs. Histone methylase inhibitors. Again, these family of drugs, they modify chromatin, and if we'll have time, we'll talk about chromatin as well. Again, another drug that modifies DNA methylation state. And as I said, there are drug screens in order to find a good combination that will improve the reprogramming process or that will replace the genes, the proteins. Okay, finally, as I said, iPS cells were only able to generate chimeric mice and not mice that all their cells have been produced from iPS cells. And later, in 2009, so that's a year and a half ago, 2 letters appeared in Nature where they actually showed that this is indeed possible. Adult mice generated from induced pluripotent stem cells. Okay, so 2 groups did manage to do it. It was just a matter of time until people managed. It's not exactly clear why people failed, but we know that there is a big variety between different iPS cells. And we can't really score them yet. But if you try it enough times, you will find a line that is probably more identical to embryonic stem cells than other lines. And these lines, as you can see, make mice. So almost all these problems have been solved. And now we can basically take a fibroblast, from a skin biopsy. So any patient that goes to the clinic, it's a very easy procedure. You can take some cells from the skin, you can expand them a few times, and then you can transduce them with 3 factors. You don't actually need c-MYC, OCT4, SOX2, KLF4. You don't actually need viruses. You don't need integration into the genome. And you can make iPS cells. So you have these pluripotent cells derived from this particular individual. And now you can, in theory, differentiate these cells into whichever cell type you want. Now, let's go back to the title of the lecture. And this is pluripotent models for neurological disorders, or in our particular case, neurodegenerative diseases. And neurons are very, very difficult to obtain. Okay, so if I am working on a neurological disorder that is affecting the brain, the central nervous system, I have a problem. Firstly, because unlike skin, which I can go to a patient and biopsy the skin, I can't really biopsy the brain. Brain biopsies are rare. They happen. I mean, there are neuronal biopsies, but they're not that common. They're rare. The other problem is that neurons, unlike skin cells, liver cells, other cell types, blood, they do not divide. They are post-mitotic. Means that if you have neurons, what you have is what you get. They don't self-renew, they won't make a lot of material. So the amount of material that we have is very limited, is rare and limited. And therefore the solution of taking pluripotent cells, differentiating them into neurons, is a very elegant solution, because we can, as I said, allow these cells to self-propagate, perpetually, and generate millions and billions of cells, and then differentiate them. So now we can generate a whole battery, a whole array of neurons, and study them in a dish. So if we derive iPS cells from individuals that carry a disease, we not only have neurons to work on, but we have neurons affected with the mutation. Okay, so this is again a very powerful tool to study the disease, because we can now generate the cell type that is affected. For example, let's go back to the example I talked about earlier, Huntington disease. We can take these cells that carry this long polyQ stretch and generate iPS cells from skin biopsies. So, short background about Huntington disease. As I said, firstly, it's a polyglutamine disease, polyQ, which means that there is an expansion of glutamine. So as I showed you in the previous slide, in healthy individuals, you have 15, 20 glutamines, and in the mutated gene inside Huntington, you can have up to 150, 120, 100. The longest known so far is 180 glutamines. There are about 11 proteins in our body that have this polyglutamine in every one of us. But normally, in the normal proteins, it's a limited length of, as I said, about 15 to 20 glutamines. However, in the mutated individuals, this length can reach hundreds. Okay, so Huntington disease is the most widely known polyglutamine-related disease. It's still a rare disease. It's about 5 for 100,000 patients in Europe. And the striatum is mostly affected in Huntington disease, although after the striatum, other brain parts succumb to the disease as well. The Huntington protein, which is, as I said, mutated in Huntington disease, is involved in many biological processes, And the truth is that we do not know why the striatum is affected. In fact, huntingtin is expressed in all the cells in our body. So we don't really know why the brain is affected to begin with, because it's mutated in all the cells in our body. And we don't really know why only the striatum in the brain is affected. So this is something that we need to find out. Here's an example of a sample from a patient. This is, are you familiar with PCR? No? Okay. So I'll just explain briefly. This is a genotyping. What you can see here is, I'll show you, is a part of the gene. So we're basically looking at a polyglutamine. Okay, so this is the so this is a wild type, and what we're trying to find out is the length of this polyglutamine, and this ladder is actually the length of the polyglutamine. Okay, so what we're visualizing here is actually the DNA that contains this polyglutamine stretch. So this, here you can see a faint band. This is a wild type. Okay, so in a wild type you have this 100. 100 is because we're looking at DNA and not the protein. So DNA, glutamine is CAG. So we have 3 letters. And we're actually looking at stretches before and after this polyglutamine. So we have about 100 bases in the wild type. So this is the wild type, and we have 100 bases. But in the mutant, we have 2 bands. Why 2? Because it has one wild type allele and another allele which is mutated. So the sick individual has one wild type allele. So the wild type has 2 wild type alleles, but the mutated one has one allele with a very long polyglutamine. In fact, we can calculate how many glutamines there are here. So we divide by 3. 600 divided by 3 is about 200, and you have to reduce these 20 or so or 100 or so bases from each side, and we know that this stretch is actually 180 glutamines. This is the, as I mentioned earlier, the longest polyglutamine stretch that have been identified so far in Huntington patients. And there is a reverse correlation between the length and the age of onset. So if you have a smaller length, you will develop the disease later in life. This sick individual with 180 glutamines developed the disease at the age of 6. This is the earliest onset that is known and the longest polyglutamine stretch. Okay, so this is called juvenile Huntington disease. It's rare, very rare. Okay, so here you can see iPS cells from Huntington disease patients, which actually we derived in our lab, and these are just markers for pluripotent cells. So these markers are not expressed in fibroblasts, are not expressed in skin biopsies, only in embryonic stem cells and only in iPS cells. So here you can see a colony. Here you can see the staining. It's a specific staining for alkaline phosphatase for pluripotent cells. This is the green staining, OCT4, one of the pluripotency factors, another red staining for another factor. So you can generate these iPS cells from biopsies, skin biopsies of patients. And further on, you can now differentiate these iPS cells into neurons. So this is just a sort of a proof of principle that we can do it, and we can generate essentially as many neurons as we like to study disease manifestation. This can be used for various different downstream applications, such as drug screening, to study the basic biology of the disease, try and understand why striatal cells are affected and not others. And what we're trying actually to do now is to generate not neurons, but striatal neurons. And this is a particularly difficult challenge to get a specific subtype of neuron. So some neurons are more easy to get, but some neurons are very, very difficult to get, and this is something that we're trying to do.
Which kind of neurons are there?
There are probably...
And the stratum are to inhibit the signals from the motor cortex, right?
So the striatum, yeah, it's true that also the striatum does not contain a single type of neuron. The striatum contains TH neurons, dopaminergic neurons, as well as GABAergic neurons. So these are the two main neuronal cell types that you have in the striatum.
Are
And
they all affected? All of the striatum cells are affected, or just certain?
yeah, that's another good question. So basically, in the striatum, you can see that there are two answers. First of all, not only neurons are affected, so there are also glia that eventually gets affected in the striatum, and also there are motor neurons that get affected around the striatum and in the motor cortex. In the striatum, we see that both cell types are affected, but probably the GABAergic are worst to begin with. So once you derive iPS cells, because you cannot generate mice or chimeric mice in humans, with human cells there is one pluripotency test that one can do, is basically to transplant these cells inside a SCID mice. SCID is severe combined immunodeficient mice. These mice have compromised immune system, so they don't reject the cells that you transplant, and then you can generate teratomas. Teratomas are benign tumors in which you can find differentiation of the cells you transplanted. And what you're looking for are basically evidence for the three germ layers. So ectoderm, mesoderm, and endoderm. Suggesting that the cells that you transplanted are really pluripotent, generating different cell types of all germ layers. So this is a pluripotency test. Another interesting disease model that we're working on is called Machado-Joseph disease, or MJD in short. It's another polyglutamine-related disease. It's rare. It's one in every 100,000, although in Portugal it's 1 in 10,000, and in the Azorian Islands 1 in every 140 individuals is affected. And today people believe that the disease originated in the Azorian Islands. So the Azorian Islands, they belong to Portugal, off the coast of Portugal, and this is where the disease is most pronounced. There's also a pocket of the disease in Israel. All known patients are actually of Yemenite descent. So it's quite interesting to understand why. So this is a spinocerebellar atrophy, which means that in this disease, in Huntington, striatal neurons are mostly dying. In MJD, these cells are mostly dying. The cells that are in the area of the spinocerebral region. And the gene involved is called SCA3 or ataxin-3 or MJD. And again, it is involved in many cellular activities. And again, it is widely expressed, and we don't know why only this region is affected, or why it starts in this region. So these are open questions. So the first demonstration that iPS cells can actually be used for therapeutic purposes, for therapy, came from the lab of Rudolf Jenisch, paper published in Science 3-4 years ago. And I just mentioned Rudolf Jenisch earlier. He received the Wolf Prize together with Shinya Yamanaka this last Sunday. And what they showed here is the treatment of sickle cell anemia mouse model with iPS cells. And actually the first author, Jakub Hanna, is now at the Weizmann Institute. So here's what they've done. They took a sickle cell anemia model mouse. Okay, so this mouse is very sick. They took a skin biopsy from this mouse, actually from the tail tip. Okay, so they took fibroblasts from the tail. They harvested them. They grew them in a dish, and they made iPS cells. Okay, so they infected them with OCT4, SOX2, KLF4, and they made iPS cells. Now they've done the important part, which is they corrected, they genetically corrected the mutation. Okay, they replaced the mutated gene with a wild-type gene. So now they have, instead of mutant iPS cells, they have wild-type iPS cells that were derived from the original mouse. And here these are the wild-type iPS cells. They differentiated them through these embryoid bodies into hematopoietic cells, into blood cells, and they transplanted the cells back into the sick mouse, and the mouse recovered. So this was the first demonstration that iPS cells, in theory, in the future, can be used for therapy. And in the, yes?
Why is that the case? If sickle cell, even if you implant new stem cells into the mouse, you still haven't changed or have you, or replaced the cells that were already present that genetically were causing the sickle cell anemia.
Right, absolutely. And in fact, this mouse is dependent upon continuous transplantation of the cells. Yeah. So we didn't heal the mouse. We have it's a kind of therapy that can maybe sustain its life.
Compared to like insulin, like giving it's
Exactly, something like that. So you can actually keep this mouse alive until it would die from old age or something like that. So, but it's just, again, it's a proof of principle that this actually works. But in theory, there's no reason why you would not be able to do it with a person that is suffering from Alzheimer's. Take a skin biopsy. Well, Alzheimer's is different because it's not a genetic disease. But nonetheless, let's take the Alzheimer example. If you take a skin biopsy, you differentiate it, you reprogram into iPS cells, you different the iPS cells into neurons, and now you can transplant it back into the patient. So it's kind of the same, because we know that the cells are still sick, but if you're going to give this person another 10 or 20 years, that's probably all it needs. So I think that there's a lot of hope behind this. A few more examples from recent literature. So this is again the Jenisch group, Cell 2009. They produced iPS cells from Parkinson's disease patients without viral reprogramming factors. Okay. Paper from Clive Svensson and Jamie Thompson. Jamie is the person who, for the first time, generated human embryonic stem cells in '98. Jamie Thompson. So they show here in an article published in Nature in 2009, iPS cells from spinal muscular atrophy patient. And the importance of this paper was that they actually, yeah, there's a figure here. We're not going to go into all the details, but the idea was that they can take these cells and treat them with a drug that is known to ameliorate these symptoms, and they show that they can get rid of these nuclear inclusions and show that they can actually manifest the disease in vitro and also the therapeutic applications in vitro. So this was a very interesting example. Another prominent scientist, Belmonte, they show disease-corrected hematopoietic progenitors from Fanconi anemia induced pluripotent stem cells. This is actually another interesting proof of principle, because these fibroblasts were not able to reprogram into iPS cells. So usually when you have a disease, you know, you can take the skin biopsy and reprogram the cells. But these cells failed to reprogram to begin with. They took patient fibroblasts and they did not reprogram. They thought that it was likely due to genetic instability. What they did was they added a wild-type copy of the Fanconi anemia gene into the mutated fibroblasts, and they now were able to successfully reprogram them into iPS cells. So this can also happen. And importantly, not only did they manage to produce iPS cells, now the disease-corrected iPS cells differentiated efficiently into healthy hematopoietic cells. So again, a proof of principle that you can, in theory, correct human cells, generate the affected cell type. Of course, you stop here when it comes to humans, but in the future, hopefully, people will be able to transplant them back into humans. This was published about 10 days ago, 2 weeks ago, and this is the first demonstration that you can model, and this is why it's so important, again, in nature, that you can model complex diseases with iPS cells. Okay, so far, people concentrated on genetic diseases because the system is complex as is. Let's start with something that at least we know the mutation, at least we know what to correct, at least we know where to look for. Here they took schizophrenia. So there is no single mutation that affects or causes schizophrenia. Schizophrenia is a multivariate disease. And what they show in this paper is that, first of all, they can produce iPS cells from human schizophrenia patients. So here you can see SZZD. SCZD is schizophrenia, short for schizophrenia. These are the wild-type controls. So they derived iPS cells, they produced neuronal progenitor cells, and they differentiated them into mature post-mitotic neurons. This is a post-mitotic neuronal marker, MAP2. So you can see here in green, beta-tubulin 3 is also a neuronal marker. So they produced viable neurons, but not only that. Importantly, they show that these cells have a phenotype which mimics schizophrenia patients. And again, I'm not going to go into the assay, just to tell you that here you can see, so this is the schizophrenia, this is the control. What you can see here are the number of neuronal processes, so there's no difference. They generated neurons in vitro that have the same numbers of neuronal processes, but here you can see functional synapses. Okay, this is a specific staining for functional synapses using the rabies virus. And here you can see that in the control, you have the same density of neuronal processes, but many more functional synapses than in the schizophrenia patients, which again is a very nice manifestation of what you actually see in the schizophrenia brain. And finally, and this came out last year, and actually today the paper came out in humans. This was a direct conversion of fibroblasts to functional neurons by defined factors, and this was done in mouse. So the idea here was that these guys said, Marius Warnig, this guy was a postdoc at the lab of Rudolf Jenisch, and he established his own lab at Stanford University. And his idea was, if we can take factors that are expressed at high levels in embryonic stem cells, put them into fibroblasts, and get ES cells, what happens if we take these same fibroblasts and we now express factors that are highly expressed in neurons? So this is exactly what he did, and he managed to get direct conversion of fibroblasts to neurons. So they basically expressed 3 or 4, again, neuronal transcription factors in these fibroblasts, and they converted directly into functional neurons, which basically, if you look at their electrical activity, you see that they're basically very similar to neurons. And as I said, the paper showing the same thing in human cells came out today. I just saw it this morning in the Nature table of contents that came out today. I think I won't have time to talk about chromatin anyway, because we have 5 minutes left. So usually I have a little bit more for that part. So I think I'll stop here, and if you have any questions, or we can discuss in the remaining 4 or 5 minutes. So I'm done. Yeah.
I know that you only mentioned it as a means to say something else, but I was wondering about the process in which one of the 8 cells of the embryo is taken out to check out for mutation. Is it becoming more and more popular method to prevent genetic diseases like cancer?
Absolutely, yeah. The PGD is becoming almost standard now. Yeah, there are a few centers in Israel that perform PGD. The biggest one is actually in Jerusalem, in קוראים לו בית חולים ליד שערי צדק. Thank you. So that's the largest in Israel. And yeah, I mean, there's essentially, PGD is now so common. So in most of the hospitals around the world, you have, or every IVF clinic now is also performing PGD. So, and it's very important for basic science, because now we have more and more embryonic stem cell lines that have been derived from PGD embryos. We actually have Huntington disease human embryonic stem cells that have been derived from PGD embryos, and we are just getting the PGD Machado-Joseph disease cells. So it's a very powerful tool, I think, and it's really growing. But yeah, I mean, the procedure for couples is becoming almost standard. If you have, you know, the problem is that there are only certain diseases for which it's really necessary to do PGD. If the disease is dominant, if the disease is recessive, or, well, you'll have to have the gene, both parents, or, you know. So, but yeah, it's becoming almost standard. Okay.